if we were to do this, and we'll see that later in the course today, is that if we were to do Sanger sequencing for larger genomes like human, it will take ages, because as I said, the sequence length is only 500 bases and human genome is 3 into 10 raised per 9 bases. So you can imagine how long will it take to cover the whole genome, right? So given that the fact, these next generation sequencing technologies, these are called NGS, next generation sequencing technologies, came about almost a decade ago and revolutionized the way we sequence the DNA. It not only revolutionized the way we sequence DNA, it also revolutionized what all different new technologies that can be developed based on next generation sequencing platform. Along with this next generation sequencing, which not only generates a large amount of data, because let's say if you have to sequence a human genome, you're at least sequencing 3 into 10 raised per 9 multiplied by 100 times amount of sequence. It requires a different kind of computational approaches, different novel algorithms were developed because of the availability of a different kind of data that was not existing before. And some of these algorithms we will learn in this course, but essentially next generation sequencing revolutionized the way we sequence DNA or any other things, like even transcriptomics it has been revolutionized by next generation sequencing. So just to give you an overview of how it is done, the basic principle remains the same. We are incorporating a nucleotide which doesn't allow the DNA polymerase to move forward because it doesn't have the availability of a 3' hydroxyl group. So the technology basics remain the same, the chemistry and the way we do it changes. So in the first case, when we were doing the manual method on a gel, we were using radio labeled nucleotides. Next, when it got automated, we use fluorescently labeled nucleotides. In this case, we not only use fluorescently labeled nucleotides, we also use nucleotides in which you can on demand remove the 3' hydroxyl group and add back the 3' hydroxyl group, right? So essentially the next generation sequencing is usually to do the whole genome sequencing. You take the whole genomic DNA, you fragment it, you add what is called adapters which attach on both sides of the fragment, so that now you have a primer sequence that can bind to it. So if this, because this is unknown, right, unknown sequence, so we need to attach adapters of known sequence so that we can design primers against these adapters to sequence on both the directions. This library of large number of fragments, this is like a huge number of fragments, millions of such fragments. They are attached to a flow cell, which is one of the microfluidic devices. And what we do first is because the number of fragments or the frequency of having a particular fragment can be very different, what we do is we do what is called local amplification. So first step is to do bridge amplification, so that instead of one fragment in one particular site, there is a local amplification and all of these are identical sequences. So when we are reading this signal, the signal is amplified. So essentially this is a signal amplification step. Okay. Once that is done, right, Now you do sequencing reaction, essentially the same Sanger sequencing reaction. In this case, we add all the 4 labeled ddNTPs, which are now reversible. Then in the previous cases, when we are doing automated Sanger sequencing or the manual method, the ddNTPs were not reversible, they were permanent ddNTPs. In this case, using chemistry, we can actually make obscure the 3' hydroxyl group, or we can remove the chemical to expose the 3' hydroxyl group. So we now can add all the ddNTPs, and what we see is that in different clusters, so if I were to, in each of the cluster there is a particular uh, nucleotide that is added. Here another particular different nucleotide could be added, here another different nucleotide could be added, here another different nucleotide could be added. So here it is the yellow color nucleotide, this is the red, this is blue, this is green, right? This is what can be added. So what it allows you to do is, in manual method we were sequencing one gene at a time or one fragment at a time. Here millions of fragments that are now attached to the flow cell get sequenced simultaneously. So after one addition of all these ddNTPs, an image is taken, a digital image is taken of the whole flow cell, and then you do like add some chemicals to remove the 3' hydroxyl block, and then you repeat the cycle again, because now the 3' hydroxyl group is available, DNA polymerase can add the next one. So if you keep on repeating this and you have a particular image with coordinates defined, right, from 0 0 coordinates defined for all the whole of the matrix, if you now stack up all these images, you will be able to read each of the clusters. So for cluster 1 you will be able to read A, T, G, C, C, T, like that, because there would be first image, second image, third image, fourth image, fifth image, sixth image, and so on. And this is the way we now sequence. Now one of the question that comes is that these fragments are generally around 150 bases. That means they are shorter. As I said, mentioned in previously, that Sanger sequencing allows us to do up to 500 bases. This is lot shorter than that. So almost like 1/5 of that size. But the number of fragments and the number of times each fragments get sequenced is huge, and therefore we can still be able to assemble the short fragments and get the longer reads. So what are the advantages? So this is just a brief overview. I don't want to go into too much details of this because this is not relevant for the course at the moment. But what are the advantages of next generation sequencing? First, because it can do whole genome and we can sequence now large number of individuals, it provides higher sensitivity to detect low frequency variants, and this is something that we will focus on some later courses. What do we mean by low frequency variants? It has faster turnaround with high sample volumes. It's possible to sequence a human genome almost within a day these days. It has higher capacity. You can sequence each flow cell, you can sequence multiple human genomes, right, up to 10 to 12 at least, if not more. And because you're doing large number of genomes, you are sequencing hundreds to thousands of genes simultaneously. So most of the whole genome sequencing projects utilize this Illumina sequencing. So in India, just for information, in India, the government of India through a DBT project, which is Department of Biotechnology project, has initiated what is called Genome India Project. where we are sequencing 10,000 healthy individuals from across India and representing different ethnic groups. So this is a huge project that is being conducted and should finish soon, and this will be one of the first projects where Indian genomes will be available for analysis. And there is quite a lot of excitement about these genomes because the Indian populations are quite unique compared to other populations from, let's say, Europe and US. So essentially, the method of sequencing large amount of DNA is to take that large DNA, fragment it, sequence it, and then join them by looking for overlapping sequences. All right. Later on in the course, you will learn about algorithms that allow us to do this kind of overlapping. So this is not done manually, this is done computationally, and there are algorithms that have been designed to do exactly that, and you learn about how those algorithms work, what is the mathematical basis of these algorithms.